So hello, good afternoon, everybody. I'm extremely delighted to introduce Dr. Tamarra James-Todd. Dr. James-Todd received her BS in molecular biology from Vanderbilt University. She then completed a master's in public health, focusing in international health at Boston University, and then went on to complete a PhD in epidemiology from Columbia University. Following her PhD degree, she completed postdoctoral training at Harvard Medical School, where she focused on reproductive and cancer epidemiology, as well as diabetes epidemiology. She then obtained a faculty position at Harvard and is currently the Mark and Catherine Winkler Associate Professor of Environmental, Reproductive, and Perinatal Epidemiology at the Harvard Chan School of Public Health. And congratulations to Dr. James-Todd, who was recently promoted to Associate Professor this past July. And it's not a surprise that she was promoted because she has written many high impact manuscripts that have changed our field. She's received several grants, has a very impressive funding record from NIH, the March of Dimes, and other private foundations. And she's obtained continuous support for her work on environmental exposures, pregnancy outcomes, diabetes, as well as racial and ethnic disparities. And I'm thrilled that she's going to talk to us today about environmental justice and women's reproductive health. Thank you very much, Dr. James-Todd. We welcome you here and look forward to your seminar.
Thank you so much for the invitation to present today. I'm really excited to be with you all and had a wonderful opportunity to meet so many of you all already. And so as Jody mentioned, I am an environmental reproductive epidemiologist. I know this is a quite diverse audience. And so this is a little bit of a different style of presentation. You're going to see me kind of start off with definitions and kind of weave together the story of, how can we, across kind of multidisciplinary fields that really impact public health more broadly, how can we do our science in a way that helps to think about and consider different ways to ask the questions that we are getting at. So on that note, I'm going to start off with the definitions and kind of lay a framework for environmental reproductive justice research, and then really dive deeper into the disparities that we see with respect to both exposures and reproductive health outcomes. I'll provide an epidemiologic example, but I do think, and you'll see me or hear me say a bit about how that can translate into the areas of toxicology and other fields that you all may be studying in, and then provide some next steps. So just to make sure that we're all on the same page with respect to, environmental justice and specifically health disparities, I want to make sure that it's clear what I mean when I'm speaking about some of the terms that you'll hear me talking about. Certainly, the field of environmental justice is not isolated to these particular definitions of race and ethnicity. There's many other social and contextual factors that impact these things, but I'll be talking as an example today about racial ethnic differences as it relates to these particular exposures and reproductive health outcomes. So race is defined by the American Sociological Association as the physical differences that groups and cultures consider socially significant. And that's different than the term ethnicity, which is a shared culture, such as language or ancestry or practices and beliefs. And so these can kind of interplay into what we define as health disparities. There are many, again, definitions out there for what health disparities are. I'm defining them as not just the differences we see, but the differences that have social, economic, and environmental significance as it relates to disadvantage. So, there are many differences with respect to population health, but whether or not those differences are linked to disadvantaged groups is, really where we get into how we would define a health disparity. You saw in the title that this talk is going to be about environmental justice. So I want to make sure that we just understand what that is. And so it's defined by the US EPA as the fair treatment and meaningful involvement of all people, regardless of their race, color, national origin, and variety of other social constructs, not only in the development of a particular set of laws, regulations, or policies, but also in the implementation and enforcement of those policies. And so as a subset of environmental justice, there's environmental racism. Again, I'm going to be speaking about the social construct today of race and ethnicity. And so I just want to make sure that we have a common definition we're working with. And so this is defined as, conscious design or institutional neglect, actions or decisions that can really result in that disproportionate exposure to environmental health burdens by populations of color. And so because I'm also talking about reproductive health outcomes, I want to make clear that what I mean by reproductive justice, and really I'm going to be talking about the intersectionality of these two things. So this is defined as the human right to control our sexuality, gender, and our work as well as our reproduction. And so this definition, it comes from, ironically, reproductive justice is not necessarily coming per se from a scientific space as far as the definition. It actually comes from kind of more the advocacy spaces around these issues. So, we're all scientists and have heard of determinants of health. And so I just put this up here because, whether it's the environment, whether it's geography, kind of the places that we live, and the various health behaviors, or even social conditions and policies and their connections with genetics, it's really the interplay and intersectionality of all of these things that really can impact our health. And so when we're thinking about this across an individual's life course, these are two different populations, one being disadvantaged, another being more advantaged. And the disadvantaged group having many more of these risk factors or red arrows than the more advantaged group, which has many more protective or resilience factors. And so the question really is, what can we do about this? How can we kind of redistribute this so that there's less of this kind of widening gap, perhaps across the life course? So I think in order to do that, we can, and we do as scientists, ask these critical questions in environmental health disparities. And so we ask questions around what are the environmental factors, or in my case, the chemicals that may be contributing to these exposure outcome differences, and whether or not, what are the conditions that are disparate. We ask questions like who's most at risk as far as high exposure or high disease burden. We ask questions around where, so thinking about place or geographic region or differences. We know that there are regional differences with respect to things like, air pollution, or in the case of some of the things that I look at, PFAS concentrations for some of these chemicals that are used in consumer products. When are we, when are the time periods? I know many of you are thinking about critical windows of time. And so thinking about the prenatal period as a critical window, or maybe later life, thinking about sensitive windows of time, maybe something like pregnancy, for example. So really focusing in on when might an exposure really have the most impact on a given health outcome. And then, of course, mechanisms, thinking about what mechanisms might be at play. Are there genetic, epigenetic, are there hormonal or even immune mechanisms that could be at play with respect to this? But I think that there are some key forgotten questions when we're thinking about environmental health disparities. And so I think the questions of why do some populations have a higher exposure? Why might those populations have a higher disease burden? And is there a connection between the two? Is there a connection between the higher exposure burden and the higher disease burden? And then the critical question of, so what? What can we do about this? And I think asking these questions really can contribute to trust building within populations, particularly those that are most disadvantaged. So it helps us with relevance and trust building in understudied communities. It helps us to develop sustainable and well thought out interventions that can be both, tested within the context of, human studies, but can really also help to inform the animal studies and work that we do. And then also, kind of more broadly speaking, this can be informative at the policy level and really impact social change to improve health. So that when we ask these questions, we're getting and kind of moving away from things that we have less ability to change, I can't change someone's race, ethnicity, for example, but I can think about things like structural racism or stress or access and availability to safer products, for example, or housing and energy injustice issues, or even beauty injustice issues. And these things are indeed modifiable. And so it really can be quite informative when we start asking these kind of questions around, why might there be these higher exposures and what can we do about them? So in my lab, we really think about this as kind of a translational approach to epidemiology and health disparities. And what that means is that here is a model where the kind of upper part, for example, D, which represents environmental exposures, these are just some examples. This is not an exhaustive list at all. None of these are exhaustive list of again, these are just examples. But we spend a lot of time in environmental epidemiology and even in kind of the biological sciences thinking about, what environmental exposures, whether they're specific chemicals, might be linked to various health outcomes. And we also do spend a fair amount of time thinking about behavioral factors that may contribute to environmental exposures. And in epidemiology, we also spend a fair amount of time thinking about and describing difference and thinking about things like how social or cultural factors may impact environmental exposures or a given health outcome. But rarely are we thinking about this kind of in its whole context and thinking about all of these upstream factors. And I would beg to say that that might be really hard to do in one study. But what if we were to kind of chop it up and think about this and really think more about some of these upstream factors and what we can do to kind of reduce exposures with subsequent impact on health outcomes? So asking why, I think, really matters. We think about this in the context of kind of sociopolitical impacts on environmental health. There's things like historical redlining that can really impact things around conditions around environmental justice communities in which people live. So for those of you who do not know what historic redlining are, this is a set of policies that were implemented by the United States back in the mid 1900s that really restricted where certain individuals were able to receive mortgages and buy land, with the green communities representing kind of the safer, if you will, more privileged communities. And so green and blue represented that, and red and yellow represented communities that people of color were restricted to living in due to inability to obtain a mortgage based on US policies. So if you look and overlay this with kind of environmental justice communities, like where is air pollution, where are oil refineries or landfills, you will find them more commonly in spaces where there was more redlining or the red communities. Likewise, retailer redlining, which we speak less about sometimes, but the same thing where we see this oversaturation of fast food in certain communities, particularly lower income communities and communities of color, with fewer grocery stores or healthy food options. So these really are food deserts, basically. And then likewise, noise and air pollution, as I've already mentioned. And so what are the implications of this? In the context of, asking why, when we think about things like beauty justice and environmental health, I'm showing some images from this is a school here in Massachusetts. It's a suburban community. These are two young girls who were banned from their school because they were wearing a protective hairstyle. They were wearing their hair in braids. This essentially allows individuals with kinkier hair to not have to apply as many chemicals on their hair. It keeps it protected and safe. And so in the context of what I'll be speaking about in just a second, when you are forced due to school policies, for example, to have to wear your hair differently, you may have to apply more chemicals, and that may have real impact on your health outcomes. Likewise, skin lightening creams, which are more common in communities of color and certainly contain all sorts of chemicals that have implications for adverse health outcomes. Here, nail salon exposures that I'll talk a little bit more about in a second, as well as cosmetics and even perfume, which again, individuals who may identify as being women, or these chemicals have higher exposure concentrations oftentimes in individuals who identify as being women. Like that this is a real issue. Even recently with the Johnson & Johnson lawsuit on talc, which, as a byproduct, asbestos is in these products, and ovarian cancer with a recent settlement, and douching, which again, these feminine hygiene products contain a variety of chemicals, and both of these have been more heavily marketed to women of color. So some examples of the specific chemicals I'm talking about from those previous slides are phthalates. And many of you all know what phthalates are, so I won't spend a ton of time other than just to say these are endocrine disrupting chemicals. They are used for their ability as plasticizers. They are also used in personal care products. They can hold fragrance in to the product. So they're also used in food packaging and medical tubing and a variety of other consumer products. As I've just mentioned, there are differences by gender. And so we see that it can only show a tenth of the concentrations here for this metabolite of diethyl phthalate that's commonly used in personal care products, but women have much higher concentrations compared to men. And that's also the case for this metabolite of dibutyl phthalate, with again, women having much higher concentrations than men. This is data from the National Health and Nutrition Examination Survey, which is showing a representative population of the civilian US population. And here, where we see, again, these differences, but here I'm showing them by race ethnicity, with the red and green bars representing African-American and Mexican-Americans compared to the blue bars representing non-Hispanic whites. And again, you see these differences, again, with these kind of personal care product phthalate metabolites. And so, what's driving that difference? And back when I first started this work, a lot of pushback that I would get on this is that, oh, well, they're African Americans are, metabolizing these things differently, for example, which, as I stated at the beginning of this presentation, race is a social construct. So while certainly things can get under our skin to impact our biology, it's not the reason why we're necessarily seeing these differences. Another class of chemicals that are also found in personal care products are parabens. Here, I'm showing data again from the National Health and Nutrition Examination Survey with Dr. Antonia Califat, an analytic chemist who heads up the lab down at the Centers for Disease Control and Prevention that actually looks at and measures these things. And again, we see these, striking differences in the non-Hispanic Black population compared to the Mexican American and non-Hispanic white populations, even as young as age 6 to 11 years. And really, what is the implication of having these much higher concentrations? What's driving these differences? So the relevance of these particular kind of classes of chemicals, parabens fall into the phenols family or class of chemicals. And so we know they're associated with a variety of adverse health outcomes, ranging from early puberty to subfertility and infertility, all the way to some of the things that I spend a lot of time thinking about, like diabetes and obesity. So the question is, are they linked? So I showed some differences with respect to chemical concentrations by some of the social factors like race. And so here I'm showing you the overlay between the health conditions and things like race, ethnicity. And so we know that Hispanic and Black girls are more likely to reach menarche earlier. We also know that Black women are twice as likely to experience infertility. We know that Native American women are 7 times more likely to experience gestational diabetes, and that's about 2 to 3 times higher for Asian and Hispanic women. We know that Black women are 50% more likely to experience a preterm birth, and that fibroids occur 2 to 3 times higher among Black women. And finally, that the prevalence of pre-pregnancy obesity is about 50% more prevalent among Hispanic and Black women. And so again, kind of that juxtaposition of the original questions that I was saying to you about asking why. So again, we see these associations. We see the higher, or the association between phthalates and phenols and these health outcomes. And we know that there's an association with respect to race, ethnicity, and these health outcomes. Why is that? And so what? What can we do about it? So I'm going to give you an example based on some early work I did, and then I'll give you a more recent example. This particular example is coming from work where we looked at hair product use as the why that might be contributing to differences in endocrine disrupting chemical exposure and racial ethnic differences in age at menarche. So I got started in this research some time ago because of a set of studies that came out when I was a master's student. So this is in the early 2000s, and we, you know, I ran across a case series that really intrigued me, where girls as young as 4 months old and as old as 4 years old had developed breast and pubic hair. And the question why, like, is it this like a family history thing? What's happening? And the pediatric endocrinologist who had seen them was really interested in learning more about what exposures that may be happening in the family. And after, you know, really, you know, at questioning the family members, learned that all of the girls were African-American. And this pediatric endocrinologist said, you know, the common thread is I'm hearing you're using hair products on these children. I'm going to send these out for independent testing. And lo and behold, what came back was different. Every single product contained 3 different forms of estrogen. And they had been put into the products unbeknownst to the parents. It was not on the ingredients list. And once they stopped using these products, the pubic hair fell out and the breast regressed. I was fascinated by this. And this is also around the time that this issue of Time magazine hit the shelves. And kind of piecing this together, might this be one of the drivers, with respect to this? And the reason why was this study had come out in the late 90s, showing these very striking differences between age at menarche. And just for full disclosure, if you looked at data on age at menarche in the 1930s, it was actually the opposite, the complete reverse. Non-Hispanic white girls reached menarche at a much earlier age than non-Hispanic Black girls. That difference kind of settled out. It was almost similar in kind of mid-century. And then by late 1900s, this difference was, where 62% of Black girls had reached menarche by age 12 compared to 35% in white girls. And so really, there was a lot of discussion around, oh, this is a biological difference. But as I just mentioned, this had actually changed over time. Biology doesn't change that fast. And race is a social construct. So what was driving this? So the other thing that had happened was this ecologic study looking at magazine advertisements from the 1950s forward, and looking at advertisements in magazines advertised to Black women versus white women. So this is a magazine that was advertised to white women in the 1950s, and was saying something to the effect of, this is estrogenically rich and hormonally, it'll beneficial, it'll help reduce your aging and so on. And you actually stop seeing these ads talk about estrogen and so on in later on in the 60s and 70s among white magazines. But these are still, happening. And in fact, if you were to go into, many of our local, like Walgreens, CVS's and so on, you'll still find products like this. Hask placenta. I just went into my local store recently and still saw this. They're using sheep and cow placenta, but this is still being advertised to Black women. So this is an advertisement from the 80s, but you can still find this on the shelves. So we decided to found the Greater New York Hair Product Study, and this really required us to, figure out a way to look at hair products and see if this was associated with earlier age at menarche, of which earlier onset age at menarche is associated with a variety of adverse health outcomes, ranging from an increased risk of breast cancer, but also a variety of cardiovascular disease outcomes as well. And we had no idea how to ask about hair products at the time. This really was an understudied area. Again, if you're thinking about kind of environmental justice issues and what is understudied, what things aren't we looking at, this can be an issue. But we decided that we first had to start off with doing some qualitative research. So we did a variety of focus groups, found out what and how to ask questions about hair products. And in the context of that, we learned a lot. We learned that people chopped up birth control pills and put them into leave-in conditioner, heated it up and applied it directly to their scalp. We learned a variety of things. After we kind of, got all of this information, I'm a quantitative scientist. I was being trained as a quantitative scientist at this time point. And so I had to take this and, design a questionnaire and then test it out. So we recruited, after designing that questionnaire, recruited 350 or so women. And we really wanted to know about the usage patterns, but also what was in the products. These were the different products that we saw that people really at the time were primarily using. And so we created a hair products label book that contains over 200 different products. That was a tool that was used to assist us with data collection for each individual. And so we found that by far, product use varied by race ethnicity, with greater use of things like hair oils among African Americans and African Caribbeans. In fact, all hair product use was much more common within those subgroups of our population compared to non-Hispanic white or even Hispanic individuals. We also had the opportunity to look at the ingredients on the labels of these products, so looking for things like parabens or fragrance, which is to some extent synonymous with phthalates or even placenta. And what we found is about 50% of individuals who identified as being African-American used products that contained one of these ingredients on the backs of the labels compared to about 7% of non-Hispanic whites. As you can see, there's even variation within the Black population, which is heterogeneous. We were able to do this in New York City, where we were able to have the power to kind of see this. Of note, labels are, the US FDA does not require full disclosure of what is in products on product labels. And so, we're only able to look at a subset of things. And so, really better understanding, the other chemical content was quite important. But that being said, with our other question, is this associated with earlier age at menarche? We did see that specifically hair oil use was associated with a 40% increased risk of starting you're reaching menarche before age 12. That association got stronger with respect to if the age was cut off at an earlier age, and for longer duration and more frequent product use. While this association with hair perms was seen, just forever use, we did not see stronger use based on greater length of time or more frequent exposures, which makes sense. Hair oils are a maintenance product. They are applied oftentimes on a regular basis, meaning, either daily or frequently, as far as like multiple times a week. They are left in and placed sometimes on the scalp and not washed off for extended periods of time, sometimes weeks on end. Whereas hair relaxers are used, kind of very infrequently, maybe once every 3 to 6 months. So big differences in like how these things are used and what the implications are with respect to chemical exposures. We validated that study and saw that the associations held even in a separate study population. This is data from the National Collaborative Perinatal Project and another cohort based in New York City. And we see this twofold increased risk for reaching menarche less than age 11 years for really it's hair oil use again is the bad actor here. So why hair oils? What's in hair oils? And so when you're thinking about different types of chemicals, hair oils contain a lot of different chemicals. And this is data. We shared our list of commonly used hair products with Silent Spring Institute. Jessica Halm first authored this paper that came from our data. And it showed that basically every single commonly used hair product contained multiple forms of endocrine disrupting chemicals, including some of these seen here. Some of these have been understudied and not, just basically not studied at all, but many of them have been and are shown to have, endocrine disrupting chemical properties. So we did an additional study where we looked at hair product use and hormonal activity. This paper has been since then published in the Journal for Exposure Science and Environmental Epidemiology, where using a hormone receptor assay, we see that these products are able to bind to and either operate as estrogen agonist or antagonist, as well as also perturb the progesterone receptor, glucocorticoid receptor, and androgen receptor. So, what happens when, again, people are using these products regularly, daily? What happens when you have these types of perturbations, and, you have, things that are operating as estrogen agonists, for example, for something that someone's using on a regular basis. So on that note, I just want to transition into the next example on hair product use and endocrine disrupting chemical and racial differences in pregnancy outcomes. And so in our lab, we really spend a lot of time thinking about how pregnancy can be a stress test, not only for, looking at things like gestational diabetes, where we know that there's a 7-fold increased risk of type 2 diabetes following a pregnancy complicated by gestational diabetes, but also thinking about environmental chemicals and how those may contribute to associations and differences we see with respect to pregnancy complications, but also in later life. Here, what I'm showing you is a graph where there are 2 different populations represented. One is a population that is of high risk, and this is the red group. The lighter blue, it represents kind of a healthier population, a more normal or average risk population. Both of these populations experience 2 pregnancies. These are the blips here that are happening. And they both continue on into later life. The difference is that there's a clinically relevant threshold, in this case, because we're looking at a y-axis that represents glucose levels. Once an individual crosses this clinically relevant threshold, in the context of pregnancy, they're diagnosed with gestational diabetes. So this happens twice in this kind of higher risk population. And outside of pregnancy, in middle age, they go on to develop type 2 diabetes when they cross this threshold. Note that earlier development of type 2 diabetes typically leads to more severe form of disease with more challenges with being able to control this disease. Unlike the healthier population that also experiences 2 pregnancies, does not cross this clinically relevant threshold and has a more delayed onset of type 2 diabetes if they ever do go on to experience this. And so the question is, can we learn anything from the pregnancy itself? So can we develop targeted interventions? And there's been a lot around kind of lifestyle differences and so on that are actually able to mitigate this risk. And what might be driving this difference between the 2 populations? And so I'd like to posit that perhaps even environmental chemical exposures could be contributing to this difference in, metabolic health that actually increases and changes this kind of trajectory for this kind of red population, or this more complicated and higher risk group. And so if we learn from the first pregnancy that there's something perhaps going on and we know about their chemical exposures, might we be able to design targeted interventions, reduce the chemical exposures, and then subsequently lead to lower risk of recurrence of gestational diabetes, which is more likely when you have a pregnancy complicated by this condition, but also delay the onset of type 2 diabetes. So again, are there ways to reduce environmental chemical exposures, for example? This is another way to think about this. Again, with one population, up here having higher concentrations of these endocrine disrupting chemicals, and that this really leads to a different trajectory for cardiometabolic health. So more weight gain, higher risk of gestational diabetes and hypertensive disorders of pregnancy during pregnancy, but then also following pregnancy, a period that we tend to not spend as much time thinking about, the postpartum period. So more weight retention and weight gain, more insulin resistance, and perhaps even higher blood pressure. And then we look later on in midlife and see higher risk of obesity, diabetes, and cardiovascular disease. And for some of you all who do work in kind of the menopausal space, kind of just accelerated reproductive aging with earlier onset of menopause and so on. So kind of just thinking about what does this look like across the life course. So in pregnancy, we know that chemicals, these are non-persistent chemicals, phthalates are, and so we know that these things can vary. But one of the things I think is worth noting is that even though they may vary, the fact of the matter is that what we're seeing in our studies is that the higher concentrations are sustained. So here we're seeing non-Hispanic Blacks, Hispanics, again, variability that I think is really interesting and could maybe even help us to identify things that we can do to reduce sources of exposure. But regardless, these 2 groups remain significantly higher compared to non-Hispanic whites when we're looking at here monoethyl phthalate or metabolite of diethyl phthalate across pregnancy. When we're looking at pregnancy health specifically, as I mentioned earlier, we know that infertility and phthalates are associated with a variety of infertility-related outcomes, ranging from decrease in antral follicle count and even to an increased risk of pregnancy loss. I know folks in this group are doing a lot of work around this. We also know that there's associations with preterm birth and small for gestational age. We know that there's associations with increased risk of preeclampsia and even other hypertensive disorders of pregnancy. And in our lab, we spend a lot of time thinking about elevated glucose levels and gestational diabetes risk. So when we're thinking about this, what are there particular time periods even that these exposures may be particularly lead to these adverse health outcomes? So relevant to that and relevant to the work that I just mentioned about hair products, we decided to look at the association between hair product use and preterm birth in the ERGO study. This is a cohort study based here in the Boston area. This is pilot data from that study looking at hair oil use. Again, these kind of bad actors. We did look at other types of hair products, but we only found, again, these associations with hair oil use. And it's specifically, when you're thinking about the time window for this particular health outcome, we see an association for daily use of hair oils in later pregnancy and a 10-day earlier delivery. Now, for those of you who may not study preterm birth, there are very few things that are associated with a 10-day earlier delivery. And so this is something, again, with that 50% increased risk of preterm birth among African Americans, might this be something that is a modifiable risk factor? Might someone be able to say, you know what, maybe reduce your hair oil use, and that may be able to contribute to reducing the exposure and subsequent risk of preterm birth, for example. So this we're further investigating. So I'm going to end here with one final example around acculturation, personal care product use, and gestational diabetes. And the reason for focusing specifically in on this is, again, acculturation, in thinking about race ethnicity, there's a variety of social and contextual constructs that may be contributing to differences that we see. And as you recall, I showed a 2 to 3-fold increased risk of gestational diabetes among Asian women. So that's what I'll be talking about here. So conditions that are needed for an environmental health disparity include the following. There must be an association between the less advantaged group and the health outcome of interest. There must be an association between the less advantaged group and the environmental exposure. And there must be an association between the environmental exposure and the health outcome that we're interested in. So for this example, we're looking again at race ethnicity and gestational diabetes. And so here I'm showing you that we do indeed satisfy this difference. This is data from Kaiser Permanente, Northern California. And what you're seeing is the prevalence, age-adjusted prevalence of gestational diabetes by race ethnicity, but also stratified by place of birth. Now, when I first started doing a lot of work in gestational diabetes, I'd get this kind of hand waving like, oh, the racial ethnic differences are solely due to differences by, with respect to genetics, that there's some sort of beta cell polymorphism that delays insulin signaling that actually contributes to this, this difference that we see for Asian individuals compared to non-Hispanic white individuals. The problem with that argument is there's no way that genetics can change that quickly. So for individuals born outside the US, for example, Korean individuals have an 11.9% prevalence if they're born in the United States compared to Korean individuals who were born outside the US with a 5% prevalence of gestational diabetes. So here we see these differences, and it's quite interesting and striking. So next, association between the less advantaged group and environmental exposure. And again, I've already shown you this data, but again, just showing the National Health and Nutrition Examination Survey, we do see differences. One challenge that has, that we've had up until recently is that the Asian population has not been disaggregated from the other populations. So this is a really understudied group because we often aggregate them into the other category, or we don't do studies where we have a large enough Asian population to actually look at this. Finally, the association between environmental exposure and health outcome. And this is data from the TIDES study, and this is showing concentrations of various phthalate metabolites averaged between first and third trimester. And so this is the odds for each 1 unit interquartile range change. And here I'm showing that, again, this monoethyl phthalate, or this fragrance related personal care product chemical or metabolite of this chemical is a 60% increase odds for 1 interquartile range increase. And so, you know, what's going on? So here, I'm just noting that we've met the criteria for having an environmental health disparity based on kind of what I've laid out here for each one of these. And often the next step that we'll do, and many of us do this, we like to do stratified analyses. They're fun and they are helpful. They're descriptive. They let us know that there might be something else going on. And so we indeed did do that in this study. And so we see that only the Asian population here has associations between these phthalate metabolites and higher glucose levels as an indicator of gestational diabetes risk. And so that lets us know that, but this isn't sufficient. We cannot stop here. We would not really understand why is that. And to be honest, we kind of reify these racial ethnic disparities and differences as if they're biologically driven when we stop here. And so again, going back to let's ask the question why might there be this going on? You know, so that we can actually figure out what we can do about it. I would say that we need to transition from simply stopping at effect modification. It's very important to do these types of stratified analyses, but we may, for purposes as I just laid out earlier, need to consider and think about the role of mediation analyses in the work that we do. So if we were to do that, we may even have to consider a multiple mediator model where we really are thinking about the sources of the exposure, in this case, sources of phthalate exposure, and something that is also racially, ethnically patterned that may be contributing downstream to gestational diabetes risk. Because I can't change race ethnicity, but I may be able to change this X, this modifiable factor here. So some things that we've been thinking about, I mentioned acculturation earlier and those differences based on place of birth. Here we looked at urinary phthalate metabolite concentrations by place of birth, and we see real differences. So again, if you're born outside the US, this is looking at monoethyl phthalate, much higher concentrations in individuals born outside the US compared to individuals born in the US. And length of time matters. So you start looking more like individuals born in the US based on like longer time period in the US. Again, these are metabolites of chemicals commonly found in personal care products. So acculturation may be a driver of the disparity. Place. So, you know, oftentimes individuals, we live in, many communities, many populations are still living within ethnic enclaves or due to redlining and other issues, living in communities that are, greater, a lot higher or lower populations of color. So here, this is looking in the Boston area. We've recently done a study where we're looking at access to safer products and the availability of safer products. And we collected data on over 1,400 different products at various stores and then assessed their environmental working group risk score. With the higher the number, the worse off the product is. Going from left to right, lower income populations and higher percentage of minoritized populations, then going to right, higher income, lower percentage minoritized populations. And so you can see it, particularly for these other hair products, for example, the average environmental working group risk score is much higher compared to, in a lower income, more minoritized. This is a population of more Hispanic individuals compared to Wellesley, which is a wealthier, predominantly non-Hispanic white community. So just what's available in the stores may matter. And even with internet purchasing, it turns out that people still buy most of their personal care products from, big box stores and like Walmart, Target, and pharmacies like Walgreens and CVS. Structural and workplace factors. So here I'm showing you data from the American Communities 5-year sample survey. And what you're seeing here is nail salon worker characteristics. So more predominantly Asian, female of reproductive age, born outside of the United States. And so when we're thinking about the types of exposures that folks have, one of the things that's worth noting is that workplace exposures may be driving some of that difference that we see. In fact, the butyl phthalates were one of the, if we went back, the butyl phthalates were one of the stronger associations for higher glucose levels. Well, might it be from the workplace exposures? Because to be honest, workplace and occupation are not random. Those are also structural risk factors. So when we think about this, then it's not necessarily a stopping at effect measure modification, but thinking about things that we can actually maybe consider, intervene upon that could contribute to reduction in phthalate exposure. For example, if we know that it's workplace exposure, we can make sure that people have the appropriate ventilation or personal protective equipment that may be able to reduce their phthalate metabolite concentrations with subsequent impact on gestational diabetes risk, for example. Just to end with several examples of other work that sits at the intersection of environmental justice and reproductive justice. There's some ongoing work happening in Kim Harley's lab looking at cosmetic use and phthalate metabolite concentrations among Mexican-American girls and adolescents. Ami Zoda is doing some work in the feminine hygiene product space in phthalates and fibroids. There's a lot of work happening with John Meeker and Northeastern University, their partnership with the University of Michigan around personal care product chemicals and preterm birth among Puerto Rican women with the PROTECT study. There's a fair amount of work that we're doing around acculturation, phthalates, and gestational diabetes, working with our community partner at South Cove Community Health Center in Boston's Chinatown. And we continue to do work looking at environmental exposures in pregnancy and postpartum health, really thinking about this in the context of Black maternal health and thinking about exposures like hair products that are unique to Black populations. So my recommendations for kind of moving this work forward, regardless of where you sit, whether you're an epidemiologist or a toxicologist, is really considering the role and the spaces and places in which we look at and think about exposures that may happen in more diverse populations. Basically being able as epidemiologists, for example, to study more diverse populations, including women with concurrent chronic diseases, but also individuals who are, of racial and ethnic minorities, such as multiracial ethnic individuals and Asian individuals. Also looking at and examining individual and contextual determinants of endocrine disrupting chemicals. So thinking about the social, cultural, and policy-based determinants. It was wonderful during the day today to really speak to folks from a variety of disciplines. You all really have it all there. And so being able to work with anthropologists and toxicologists and so on, and thinking about all of these things. Recognizing that stratified analyses are necessary but not sufficient, and there are other tools that we have in our toolkit, including mediation, mixed methods, and multi-level modeling. Thinking about social and environmental interactions, and also studying and thinking about those chemicals, those environmental exposures that are less well studied, as well as the conditions that are maybe less well studied within certain groups. Because at the end of the day, we want to go from a model that looks like this to something that looks like this. And I would just like to acknowledge and thank my various collaborators and funding organizations, and I can take any questions.
Thank you very much, Dr. James-Todd, for an outstanding presentation. Really well done, and we appreciate it. I will ask the IT people to unmute, and if people have questions, please feel free to go ahead and unmute yourself or write your question in the chat. Oops. Sorry, Tamarra, I'm trying to allow people to unmute themselves. So questions for Dr. James-Todd? I don't know if people can, I'm unsure if people are unmutable.
Hi.
Oh, you are, okay, so I was able to do it. I wasn't sure if I could unmute or not. Okay, go ahead, Lori, I'm sorry.
Thank you so much for a really fantastic talk. It was great to hear. And I think gave us a lot to think about in terms of how we model our animal studies as well. I was wondering if some of the effect of hair oil exposure is due to prior to pregnancy, or if you think it's all related to direct exposure during pregnancy, that the effect on the outcome.
Yeah, that's a great question. So right now, our models, we're specifically looking at and testing pregnancy as a sensitive window. And Jody, I don't know if I should, if I have a second, like, I know you're asking about Lori about the hair products, but I do want to, maybe I can just show something that may be of interest to folks because of this kind of like sensitive window of pregnancy. So we did, Lori, ask the question about some other chemicals, the per and polyfluoroalkyl alkyl substances that maybe some of you all know. And we asked this question in human study, and then we followed it up with a study in an animal model. And let's see if I can get this to advance. There we go. So in the human study, what we were doing is we wanted to know whether or not these are persistent chemicals. So they're used as surfactants. They have like a kind of, not like a anti, water resistant and grease resistant properties. And so in looking at this, what I'm showing you is data from Project Viva, a cohort study. And you'll see here, we're looking at gestational weight gain at 1 year, and 3-year postpartum weight change. And I'm noting that I'm only looking at first trimester exposure to these chemicals. And so the PFOS and PFOA, which are the long chain PFASs, that's what they're also called, have significant associations only for 3 years out. So, in pregnancy, this still doesn't really per se answer the question. We see a similar association, set of associations for immune markers and other adverse cardiometabolic health outcomes. For in red here is the adverse effects, in green, things that we didn't expect to see, which were kind of more positive heart cardiometabolic healthy associations, again, for mainly PFOS and PFOA, but some of the others as well. So to ask the question that you're getting at, we had to do an experimental model. And so what we did here was we used a mixture of endocrine disrupting chemicals, and this is working with Dr. Marissa Terry, pictured here, where we dose the mice only during pregnancy, so not preconceptionally. And then we keep them alive after they deliver. And we do a variety of testing. And what you can see here, again, if they're only dosed during pregnancy, not pre or post, we see that there's significantly elevated glucose levels, higher LDL, and higher free cholesterol. So we do think that it's possible. I'll just show you one more thing. We also saw more visceral fat in those that were dosed only during pregnancy compared to those who that were not dosed, and that's represented here in purple. So I think pregnancy, Lori, can be a sensitive window. I know it's a completely different chemical class and so on, but there might be something going on there.
Thank you. Other people want to unmute and ask Dr. James-Todd a question?
I think Rachel has her hand up.
Okay, go. Oh, yeah, go ahead, Rachel.
Hi, thank you. That was really a cool talk. Can you hear me?
I can hear you. Yes, thank you.
Okay, great. Several times you said that that human genetics can't change that fast, and it just made me think that, well, that's true. Microbiomes definitely change on that time scale, and I was wondering whether you have any like part of what you're working on that incorporates that change as well.
We do. I got pulled. I think I was talking to someone earlier. I got pulled into the microbiome world about a year or so ago. That was one of the questions that came up with the acculturation study was, well, what about diet? We know diet is so heavily linked to gestational diabetes risk. And so, is there anything about these, chemicals, or even kind of, all the questions around length of time in US dietary practices and so on that could really be impactful to the gut microbiome, which is what we're studying. So we do have a pilot study funded by our P30 Center where we're looking among Chinese American women. They had to be born outside of the US. So we kind of restricted to that, and within the US for a certain period of time to kind of, because it's a pilot study, but we are looking at chemical exposures and gut microbiome as it relates to gestational diabetes risk. Yeah.
Cool. And what about like skin and hair with the hair oil things? Are you including that too in some other work, or?
Yeah, so we have asked questions around, so not with respect to microbiome, but we have asked questions.
That's what I mean. Yeah.
Yeah. So we've not, that's a great question. I had not thought about that, Rachel. So that's a great seed plant. But yeah, we've asked questions around and it recently got funded to look at the question of a type of alopecia. So I, as you see, mostly do work in diabetes, in the space of diabetes. But I was contacted by the National Medical Association, who was really interested in alopecia and skin disorders that were related to immune health, that these chemicals could be disrupting. And so we're looking at a type of alopecia that almost exclusively affects African-American women. I mean, you almost never see it in any other group. And it is a type of alopecia that is influenced by an immune response. And the question is, might these hair products be contributing to that? We are doing scalp biopsies, and maybe there's a way to think about, how can we build a microbiome into that? So I hadn't thought about that, but that's a great question. Thank you.
Cool. Thank you.
So I think we have time for one last question. Just scanning my screen here to see if anybody's hand is up.
I hope it wasn't too far out there. Oh, shoot.
Yeah.
I think one person unmuted. Hi.
Yeah, go ahead, Jim.
Just a comment. My wife is an elementary school teacher, and she teaches ESL to children who are new to US, or they are new to US, but they are raised by the parents who are immigrants. And one day we were having a dinner, my wife and myself, and she said, in school, the teachers use the term, I don't want to use that exact term because it's kind of be a aggressive term, but she say children from the parents who works on the nail salon, they have a very high level of chance to have some behavioral issues. So she thinks that something coming from the products impact the mother when they were pregnant, or they bring them to their home and expose their children through whatever things they carry with them. So have you seen any observation like that in your study?
There's some really nice work being done out in California that is looking specifically at the Vietnamese population. So that data that I showed from the American Community Survey indicates that it's, again, all these populations, I think oftentimes when we talk about race, we talk about, like, we say Asian, and we forget that, like, there's subsets within the population that may, right?
Yeah, I saw that 70% of the workers working the nail the business, they are from Vietnam.
Exactly. So, like, one of the pieces of work that's ongoing right now is looking at pregnancy outcomes within the nail salon worker population in California, and really thinking about the butyl phthalates, because that's the exposure, is that we saw associations with acetone, which I don't know how many people are actually looking at acetone and health effects. Many of the folks on this phone call know much more about the child health outcomes than I do, but certainly there are neurological outcomes that are associated with prenatal phthalate exposures and early life phthalate exposure. In children, we see different associations, and that's still under study and so on. But what I would say is that is rich for analysis. There's very little work focused in on Asian populations as it relates to these health effects. And in the context of thinking about how this works for, like, things like toxicology, I think it's going to be really critical that we design experimental studies that get at those differences in the different distributions of these chemical exposures. as it relates to the health outcomes. And I'm sorry that I ran over, but I would be more than happy to talk with you in more detail about that. But yeah, this is an issue.
Thank you.
No need to apologize, Tamarra. We really wanted to hear your answer to that question. So, but I do think in the interest of time, let's all thank Dr. James-Todd for an outstanding presentation. And I thank everybody for Zooming in today and spending time with us. Thank you so much, Tamarra.
Thank you all.
We hope to see you again soon. Thank you.
Thank you, everybody. Have a good
Bye-bye.
day. Bye-bye.
Thank you.